Okay, good morning. We are going to start now. Got a pretty fair number of people in, and anybody that comes in later, I'll just keep adding. So I say that every day, every time too. Nothing like getting a scam call right at the beginning of class. Okay, so today we're going to continue looking at the cells. We're going to go back and pick up at Tay-Sachs disease here. I've had issues the past 2 afternoons with Zoom connections in lab. So if the sound gets sketchy here in lecture, let me know right away. I don't know what's going on with it. I don't know why it's only been limited to lab, but we'll find out. Hopefully today's lab will work okay. I have a theory that if I reset and reboot my computer between the lectures and labs, that it seems to work because everything works in the morning. So, but I don't know. Anyway, any questions on anything?
I have a question about the practice tests. They don't count as a grade, right? We can just keep taking them over.
You can take them as many times as you want. They're there to give you a good idea of what to expect on the real test. And the topic areas are the same topic areas. And really, there are only so many ways to ask questions about the same topic. Just after a while, they all start to look the same. So I find them to be very useful.
Okay, thank you.
Welcome. Good question. Any other questions? Okay, then I'm going to go in here and see if this works. And
where exactly are the practice tests?
Oh, if they are in under assessments in eLearn, and go to assessments and then go to quizzes, and they are found below. They should be found below the regular exams. So, and they should be open from now until December the 10th, every one of them. So go assessments to quizzes, and then at the bottom of the list of the real exams, you know, because the real exams are labeled lecture test 1, 2, 3, 4, and so on. And then the practice exams are practice exams 1, 2, 3, 4, and 5, and they correspond to the, you know, practice 1 is unit 1, practice 2 is unit 2, that and so on.
Okay, thank you.
Certainly. Okay, now let's see if I can get this. Oh no, that's bad. Do you all see the slide that says lysosomes?
Yes.
Okay, good. That's a good start. So let's see if we can get this to work now. Okay, so we're talking about lysosomes where we stopped on Monday. And lysosomes, we're still looking at organelles. Lysosomes are organelles. And lysosomes contain digestive enzymes. You know, they're very powerful enzymes, and so they're contained inside the lysosome structure. Same phospholipid bilayer, but they keep the enzymes in there because otherwise the cell would dissolve it, would eat itself if it couldn't control it. It's how the cell can eat cell fragments or destroy any kind of bacteria or virus that tries to invade it, any kind of pathogen, a protozoan, a fungus, a chemical toxin, because some bacteria work by making toxins, making poisons. And we use these to break those things down. We also use them in our bones. We use certain types of lysosomes to actually dissolve bone to get the calcium. Now, if the cell becomes damaged and can't repair itself, then we use our lysosomes to actually destroy the cell, self-destruction, if you will. And so, and they look, sort of like this. They're these are structures inside the lysosomes that are being dissolved. This is a lysosome. I don't think they're really orange, but that's what they look like anyway. Now, the thing about lysosomes is that there is a genetic disorder known as Tay-Sachs disease. And Tay-Sachs is a very unusual kind of condition. See, when we are in utero and after birth, our central nervous system is developing. Our brain develops and gets bigger. Our spinal cord gets bigger. It enlarges. It grows. And there are glycolipids that grow along with it. They help to support it and provide energy. A carbohydrate with a fat attached to it. Well, to keep those glycolipids in the brain and in the spinal cord from collecting and becoming too large, we have a specific type of lysosomes that continually break this down so we can keep the amount of this glycolipid in check. Perfectly normal response, we take care of it. We need the glycolipids, we just don't need too much of it, because if we get too much of it, we start compressing the brain and compressing the spinal cord and destroying it. Now, when that condition occurs, it's usually the result of a genetic disorder known as Tay-Sachs disease. Tay-Sachs is caused by a lack of that particular enzyme that breaks down the glycolipid. So who gets it? Well, like any good genetic disorder, I should say bad disorder, you don't catch it. It's inherited. This is a homozygous recessive disorder. You have to have a recessive trait for your mother and your father to have this. Now, so that makes it much more challenging to develop this condition, because you have to have parents that are the carriers of the trait. And it turns out that the trait shows up primarily in 3 different population groups. Now, the largest of the groups came from what we refer to as Central European Jews. Now, European Jews for hundreds of years stayed in a fairly isolated, not isolated, but closed community where they married within their community. It's not like where we're having inbreeding relationships, but it's more of a if you have 100,000 Jewish people in an area, they're going to intermarry among themselves. They don't marry outside. So, and what that does though is it keeps recessive. If there are recessive traits, it keeps them alive in the population. And Eastern European Jews had a higher rate of occurrence of Tay-Sachs than anybody else. And the consequence of Tay-Sachs is that it would kill children usually around the age of 5, because their brains were developing and their central nervous system was developing, and the glycolipids accumulated and weren't broken down like they should have been. And so it would cause, it would significantly impair their intellectual capacity. It would cause blindness, it would cause paralysis, and usually kill them between the ages of 5 and 10, usually around 5. There was no treatment, there was no recovery. So now it turns out that there are other variations of Tay-Sachs that affects adolescents and adults. See, it doesn't matter then, because once the brain and spinal cord is already developed, then these developmental issues like accumulating lipids don't hurt it. But it's only in its developmental stage that causes the problem. So it's fatal in infants and young children, but doesn't have a great deal of impact on your post-adolescent and your adult. It may have some, but it certainly doesn't have the dire consequences it does on an infant. So why are we talking about this? Well, it turns out, let me go back before I finish talking. that's the primary population group that has Tay-Sachs. You know, just as an aside, Mediterranean Jews didn't have it. Mediterranean Jews, you know, in the Middle East, married everybody. They went out, they intermarried, and so the gene, while it's probably still there, buried in DNA somewhere, has become so rare because it's a recessive trait, and the more you increase the gene pool, the less likely that recessive trait's going to show up. Now, second population that had this, and this has a point, it's getting to us now, are the French Canadians that lived in Nova Scotia, Newfoundland, and those areas back in the 1700s, 1800s, whatever. Well, they were French Canadians and they were Catholic. Okay, so now what does that have to do with anything? Nothing, except that the Canadian government didn't like Catholics. So, well, and on top of this, they had this strange disease that killed their children. So the Canadian government, which was probably England running it at that time, because after France left and then England took, anyway, it's long, we don't need to go into history. A large portion of those French Canadians that had the Tay-Sachs gene migrated to southern Louisiana, because that at the time that was still French territory. So they migrated to southern Louisiana and became known. They collectively called themselves Cajuns. Cajuns, you've heard of Cajuns. Now, what does that still have to do with us? Well, they still have a higher instance of Tay-Sachs in their population there, because Cajuns tended to keep a somewhat closed society. They stuck to themselves a lot, and they migrated north occasionally, particularly like 20 years ago when Hurricane Katrina smashed into Louisiana and uprooted large hunks of population and moved them north. So the odds of seeing a child with Tay-Sachs and a lot of them came to East Tennessee. So the reason I had this long involved discussion on Tay-Sachs disease is there's a reasonable expectation that you may encounter infants that have this condition or parents that are carriers. And normally we would expect to see this in the Northeast. You know, a healthcare provider in the greater New York area likely sees this every several times a year if they're involved with treating children, because of the large population of people there that are descendants of Eastern European Jews. Around here, not so much, but we have an influx of people from Louisiana who are descendants of the French Canadians, and they bring that gene with them. So it's something to consider. It's not out of, it's very possible that you would encounter something like this in healthcare. It's an obscure disease. It's a very obscure disease. It doesn't affect very many people, and it's it's a genetic disorder. It has to be inherited. But we have greater odds of seeing it here than most places of the country. Okay, that's just my long rabbit hole in that. Okay, so that covers most of the organelles inside the cell. Now, the cell also has a skeleton, not like ours, not a bony skeleton, but a fibrous skeleton, usually made of proteins or keratin that hold the cell together, that give it its shape. I mean, why do you think, how do you think a nerve cell can be 4 feet long, and have its very distinctive shape? Why do you think that a red blood cell looks like a squishy sack and holds that shape? It's very flexible. It can squeeze through tiny blood vessels. It's because of its skeleton. And the filament, the structures that make this up are either what we call microfilaments, intermediate filaments, or microtubules. Now, the microtubules are in a circular pattern. We see them with the spindle fibers all the time. We see them We see them with connections throughout the cell membrane, microfilaments. We see these microfilaments in our contractile muscle. We also see them in cytokinesis. We see them giving the great flexibility and strength to our cell membranes. And then we have the intermediate filaments that we use for waterproofing, for example, keratin fibers that waterproof our skin and help to make our hair and nails are made up of these tiny little filaments, if you will. We also see two structures known as the centrosome and the centrioles. The centrosome is the center of the cell, and it's a physical location where we see these things that are called centrioles. Now, the centrioles, and this is one of those hard-hitting science questions that you're never probably ever going to get asked again. But the centrioles are shaped like little barrels, sorta, and they usually occur paired at right angles to each other. And what they do is they produce the spindle fibers that go out and can attach to the chromosomes. They can they can push the cytoplasm. They allow the physical separation of, we've copied the nuclear material, and now we're pushing it and pulling it to opposite sides of the cell. And that's what the centrioles do. They organize the movement of the nuclear material and the organelles during mitosis. They organize that movement from side to side. They also form cilia. Now, most of us have cilia in various locations. We all have it in our respiratory tract, in the trachea and in our nasal cavity. It shows up in the fallopian tube, the uterine tube, to push the egg along because the egg has no way of moving. It shows up in the structure of the flagella on the sperm cell. So we have these tubules in a variety of locations, and usually everything directed from the centrioles. Another thing that happens is the cell assumes a unique shape on the edge of its membrane. The membrane will fold. The membrane will fold and create villi. Villi means fingers. I guess it's Latin for fingers. And then the villi will have microvilli. And the microvilli stick out, and between the villi and the microvilli, they're increasing the surface area of the cell membrane. And so you have, for example, our small intestine, which is about 20 feet long, about as big around as your index finger all the way through. But if you were to take, so it's like a small garden hose. If you were to take that structure and lay it out flat and stretch it out to one cell layer thick because of the absorbing area of those simple columnar cells, you would be able to cover a tennis court with to one cell layer thick. So you would increase the surface area. These villi and microvilli increase the absorbing surface area of the small intestine to this massive structure. So food and water and other materials can go right across the membrane of the simple columnar cells into the blood supply and go to the liver for detoxification. So we see this is a very unique arrangement that we find on some cells, particularly in small, where we have to have absorption and the movement of materials across a membrane. Now, the organelle that we haven't touched on yet is the nucleus. The nucleus is the largest organelle. It contains our DNA. Our DNA, of course, is our genetic makeup. DNA is contained in the nucleus, it never leaves the nucleus. It uses the other nucleic acid, RNA, as it to do its messenger work. The DNA controls the production of all of our proteins that make up our body. Most cells only have one nucleus. Skeletal muscle cells have multiple nuclei, but that's only because when we were developing skeletal muscle in utero, our skeletal muscle cells consist of lots of smaller cells that fuse together, and we kept the nucleus. Those cells don't reproduce, so we don't really need to worry about copying that DNA. Red cells don't have a nucleus. Red cells kick their nucleus out while they're forming so they can have more room to carry oxygen. As a result, red blood cells only last 4 months, and then they break apart, usually in our spleen, where we then recycle the iron. So most cells have one nucleus. A few types of cells, like skeletal muscle, have multiple nuclei, and the red cells alone have no nucleus. And the nucleus looks a lot like, the cell proper. It has its own membrane structure called the nuclear envelope, which is a phospholipid bilayer, but it is penetrated by lots of openings called pores. Within the nucleus, there is a structure called the nucleoli, which is where the nucleus makes the other nucleic acid, the ribonucleic acid. And then in the nucleoplasm, which is the counterpart to cytoplasm, we have what's known as chromatin or chromatin. And the chromatin is the DNA unwound, wrapped around a protein called histone. See, DNA strands are very microscopically small, and they are about 6 feet in length, and they're very fragile. So they wrap themselves around this protein called histone for strength and support. And so that combination when it's unwound, is called the chromatin. The membrane, of course, is of the nucleus is a double layer. It's a phospholipid bilayer, just like the cytoplasm, the cell membrane. It has a mesh over it to give it structure and support. It doesn't have to give like the cell membrane itself does. As I said, it has these pores that open directly from the inside of the nucleus right into the rough ER. They're continuous. So you go out of a pore, you leave the nucleus, and you're right in the rough ER, so you can get to the ribosomes. And then within the nucleus itself, again, is the nucleoli or nucleolus, where RNA is assembled. And this is what, a close-up of it here. This is the nuclear membrane here. You can see these little round structures are the nuclear pores. This is a close-up of the pore here. It looks sort of like a gap junction. In many ways, it very closely resembles it, because it's really just a passageway through the membrane. And this is the lamina, the filaments that form the structure here to give the nucleus its defined shape. And you see how the pore right there opens directly into the rough ER. Now, the chromatin is a mixture of protein and RNA and DNA. It's about 30% DNA and about 60% histone. It wraps around the histone. The DNA wraps around the histone and looks very much like the stripes on a candy cane. The units here are called nucleosomes, which is the DNA around the histone. The chromosomes occur when the chromatin condenses. When it condenses, then it's called a chromosome. But when it's uncondensed, we simply call it a chromatin or chromatin. Anyway, so the chromosomes only show up when the chromatin condenses. Otherwise, it's all strung out in the nucleoplasm. And it looks, as I said, looks something like this. This is the chromatin here, and the DNA is in red, and the histones are in blue, and they're wrapped around here like a candy cane. And when they condense into this mass, and we have chromosomes. And this only occurs when when the cell goes into mitosis, because other than the cell, otherwise the DNA and histones are wrapped around each other and they're strung out in these long strands, unwound. I mean, when I say unwound, I said the strand is not condensed. The DNA and the histone stay together all the time. They're either in an unwound state, still wrapped around each other. They're just not condensed into form a chromosome. And it's very important for the cell to follow a set pattern here. Mitosis makes up just a very short period of a cell's lifespan. And up until we get to mitosis, the DNA is going to stay wrapped around the histones, all strung out all over the place inside the nucleus. The cell cycle has a very brief stage of mitosis, followed by cytokinesis. 99% or more of its time is spent in interphase. Now, I know that at some point, we probably all learned mitosis has interphase, prophase, metaphase, anaphase, and telophase. I remember being told, when I was even an undergraduate, that the stages were, those are the stages of mitosis. But interphase is not mitosis. Interphase is when a cell is busy doing cell stuff. And that's the normal activities. It's only when the cell is ready to undergo mitosis that we start seeing the activities involved in making daughter cells. And that represents less than 1% of the cell's lifetime. So if you look at a typical illustration of the cell cycle, you get the impression that 1/4 of the cell's lifetime is spent in mitosis, and that's not the case. If a cell lives for 24 hours, the amount of time spent in mitosis is probably under 60 seconds from start to finish. But once we enter the mitotic stage, we go through mitosis and cytokinesis, and we get 2 daughter cells. So interphase, where most things happen to the cell in its lifetime. I'm a liver cell and I want to be the best liver cell I can be, and I want to do all sorts of liver cell things. And in the last 30 seconds of my life, I'm going to reproduce and produce 2 daughter cells, which are identical to me. That's what a liver cell, if it could think, would say. Interphase runs 99% of the cell's lifetime. It's broken down into 3 parts, 3 what we call subroutines. The first phase is the G1 phase, or GAP1. Cell's busy growing. It's a daughter cell. It's 1 or 2 daughter cells. It has just undergone mitosis and cytokinesis, and now it's growing and very active. Its metabolic rate is very high. It's churning out proteins. It's doing whatever it's supposed to do. And we go all the way from the G1 stage to what we call the S stage. Now, interphase is running in the background the whole time. In the S stage, we will replicate our DNA in anticipation of ultimately undergoing mitosis. Remember, mitosis is just nuclear replication. Creating 2 daughter cells with identical genetic material, an identical nucleus in each daughter cell. So it's not, mitosis is not cell division. It's just copying the nucleus. or as we say, nuclear replication. Now, in the S stage, we copy our DNA to make sure we have it ready to go for mitosis. The S stage happens near the end of the cell cycle, because you don't want to have a second batch of DNA floating around inside your nucleus. A lot of cells never get there. A lot of cells never get out of the G1 stage. We say they're in G sub 0. Muscle cells don't reproduce. They're in G sub 0. Nerve cells are in G sub 0. They don't reproduce. Cardiac muscle cells don't reproduce. They are in G sub 0. Damage to a heart muscle cell results in scar tissue. It doesn't repair itself. Damage to skeletal muscle tissue results in scar tissue. It doesn't repair itself. Damage to the neurons usually results in permanent damage to the in the nervous system. And we'll learn later on that it depends which nerve we're talking about. Inside the brain and spinal cord, damage results in damage with no repair. Inside the nerves that lead into the central nervous system, damage can be repaired as long as it's confined to the axon of the neuron. The axon is the long extension. Because if we couldn't repair damage to nerves outside the brain and spinal cord, how could we ever do transplants? How could we transplant tissue? How could we reattach fingers and arms and legs and feet? You know, how could we do any kind of transplanting if we couldn't reattach the axons, or if the axons wouldn't grow back together again of a nerve? That only works on the nerves that are outside the brain and spinal cord. Brain and spinal cord are the central nervous system. Everything else is what we call the peripheral nervous system. So those nerves out there don't reproduce, but they can at least repair themselves. Anyway, so you either are a reproducing cell or you are stuck in G sub 0. You never get out of that. So essentially, the skeletal muscle cells that we have now are the ones that were formed in utero and we were born with. The same with our nerve cells. They were the same ones that were created in utero and we were born with. They are as old as we are, and the same with our heart muscles. Our heart muscle cells are the same cells that were formed 3 weeks after conception. You know, from the time the egg is fertilized by the sperm cell, 3 weeks later, we have a beating heart. You know, we're maybe a half an inch in size, maybe a half an inch. 3 weeks later, we have a beating. You know, 3 weeks after conception, we have a beating heart. That heart will continue to beat now for the next 70, 80, 90, 100 years. Those cells, though, that are forming the heart don't ever reproduce. The cells we have at birth in the heart are the cells we have now. They can get bigger and stronger, just like skeletal muscle can get bigger and stronger, but we're not going to add to it. So all those cells are stuck at that same stage, same stage in interphase. Now, if you are a reproducing cell of any kind, one that will grow new ones like bone cells, for example, or epithelial cells, when we get to the S stage, which comes right after G1, then we'll make a complete copy of our DNA. And we take our DNA and we use it, our original DNA that we have with us in our cells as the model to build two copies of it. And then we go into the G2 phase, where we're busy making proteins and enzymes that we need to complete the process of mitosis and then ultimately cytokinesis. So this is a cell cycle. This is what it looks like. Interphase runs all the way around to the mitotic stage. Now, in reality of timing, this entire phase of mitosis and cytokinesis is about the equivalent of this line right here. That's about how much time the cell spends in that phase. So it's not 1/10 of its lifetime. It's about 30 seconds. Now the synthesis stage where we make a copy of the DNA takes about 2 hours because you got to assemble somewhere in the neighborhood of 10 billion nucleotides. The nucleotide is a phosphate attached to a 5-carbon sugar attached to a base, a nitrogen-containing base. So, and to make to make the 10 billion nucleotides you need to assemble your DNA strands takes about 2 hours. So, but that's still running in the background while interphase is still going on. The cell's busy doing what it has to do while this other thing is going on in the background. Even in G2, interphase is still running while this is going on in the background. These stages occur very near the end of the cell's life cycle. So, how do we copy our DNA? Well, we use a process known as semi-conservative replication. The next slide will give you that name. To make sure that we have a perfect copy of our original DNA strand. Our DNA is a double helix, and each strand's about 6 feet long. We have lots of these strands embedded inside our chromatin. What we will do is we will open up the DNA strand. The DNA looks like a spiral staircase. Imagine, if you will, you could split the staircase in half. You have a half a step on one side and a half a step on the other side. And then you come along and rebuild the missing portions. Now you have 2 spiral staircases. That's essentially what's happening here. We take apart our DNA strand and we use the original strand from each side as the model to build 2 brand new strands identical to the original strand. We use an enzyme called helicase, which opens up our original DNA strand. The strand we have in the nucleus of the DNA we have in the nucleus of our cells, when we're going to copy it, we split it in half. And using that as the model, as a template, we build back and end up with 2 strands. Where it opens up is called the replication fork, because it looks like a fork here. You see how it spreads out like this, like a fork in the road. And as we open that up, we're not tearing it apart from the rest of the DNA. We form a bubble. This is a bubble. This is what we call the replication bubble. And so the helicase opens up these bubbles in the DNA strand. And then at the side of the bubbles, we start building the other half of each of these 2 strands. And we end up with 2 brand new DNAs, identical. We use something known as DNA polymerase. It's an enzyme that makes DNA, a DNA polymerase, an enzyme that makes DNA. It assembles the nucleotides that are complementary to the template, to the model, to the original strand. If the original strand had an adenine, then you're going to attach a thymine to it. If the original strand had a guanine, you're going to add a cytosine to that. You're going to build the new strand, the new DNA, using half the old one. And so we have identical daughter nuclei, DNA molecules, based off the original. So we have very, very few mistakes in here. This is why we call it semi-conservative replication. Each new strand consists of, it's half old strand and half new. We use the old strand as the model to build the complete strand. And so we can produce 10 billion pairings in about 2 hours with about 10 mistakes, 10 errors, that's all. The error rate for semi-conservative replication is 1 in a billion, which is phenomenal. 1 in a billion. Yeah. So the world's population is 7 billion people. So 1 in a billion would be 7 people. You know, if you can find 7 people out of 7 billion, that's the error rate. There's nothing wrong with those 7 people. Just saying 7 people out of the world's population represents 1 in a billion. So 1 in a billion is a phenomenal rate. We use the DNA polymerase to start attaching these complementary bases to each other, guanine to cytosine, thymine to adenine. We build this along here. As we start building the fragments of DNA, we glue it together using ligase. We use helicase to open it up, ligase to put it back together again. Semi-conservative replication. Without the DNA polymerase, we can't do it. The error rate is pretty remarkable because all we're doing is we are assembling the complementary bases to the original strands. And as I said, but now sometimes there will be mistakes. Sometimes an adenine will get paired to a guanine, or a thymine will get paired to a thymine. Those mistakes are usually hunted down. There are enzymes that slide along the DNA strand looking for mistakes. And when they find them, they will cut them out and replace them with the correct nucleotide bases in place there, if they find them. Sometimes, and in fact, many times, it doesn't matter because the error rate, the mistake with placing the wrong nucleotide base on the DNA strand may not cause an overall error. I'll show you that later on here. But with an error rate of 1 out of a billion, that's a phenomenal number there. Originally, before we cleaned up the mistakes, the error rate was 1 in a million, which is considered pretty high. An error rate of 1 in a million is reduced 1,000-fold by cleaning up the mistakes and taking it down to 1 in a billion. Anyway, this is semi-conservative replication. It does a remarkable job in giving us 2 sets of DNA now in the nucleus that are identical. They are absolutely identical. So this is how we do it. We use a template strand and we build the new strand off the template. And we do it on, for each, and we split the strand and we use the old strands as the templates. And we end up with 2 identical DNA molecules in the nucleus. This is the end result of the S stage. Now, after we get our DNA copied, and we get our organelles copied, and we get our enzymes and proteins ready to go, we can move into the mitotic phase of the cell cycle. which is very, very brief. We have mitosis and cytokinesis. If we don't undergo cytokinesis, we end up with a cell that's really big with 2 nuclei. If a cell underwent mitosis and failed to divide, you would have 2 nuclei in a big cell, and it would probably be non-functional. The cell cycle is very tightly controlled. Cells should only be dividing when they need to, when they're worn out, or when they are, replacing damaged tissue, like in epithelial cells, for example, and not divide randomly. Uncontrolled cell, cell growth is the definition of cancer. Cells that grow and reproduce and grow and reproduce without any checks and balances on them. There are no controls on them. That's what a cancer is. We don't like that. It's unnecessary. So these videos show real time just how rapidly the stages of mitosis and cytokinesis occur. You see how quickly the cell physically separates in cytokinesis. Not this one, there we go. This one actually shows the stages from just past metaphase. Here it comes again. Just past metaphase up to telophase, where the chromosomes have lined up in the middle of the cell till they've actually migrated across. And if you watch that one again, you'll notice that cytokinesis is just starting now. Cytokinesis actually starts in anaphase. So this is exactly how fast the actions of mitosis and cytokinesis take place. So if the cell cycle illustration appears to show 1/4 or 1/10 of the time that the cell's alive, that's not reality. The reality is seconds for this to occur. So now the stages of mitosis, of course, are prophase, metaphase, anaphase, and telophase. Now you may hear terms like, prometaphase or prophase 1, prophase 2, metaphase 1, metaphase 2 in other books. But we're going to stick with the basics because we just want to cover the 4 key topics here: prophase, metaphase, anaphase, and telophase. Now remember that we don't have a cell wall. We just have a cell membrane. Plant cells have a cell membrane too, but they need the cell wall to protect themselves from water loss. We have a cell membrane, but our cells are protected inside the integument, our skin. So, but the process is exactly the same, other than the cell wall here. The process of mitosis in plants and animals, in fact, in everything, is essentially the same. So prophase, what happens in prophase? Well, the chromatin condenses. And why is it condensed into these chromosomes? Well, because each DNA strand wrapped around the histone is 6 feet long, and it's microscopically thin. If you were to try and pull those strands from one side of the cell to another side of the cell, there is real potential there for damage or getting twisted around other DNA, other strands of DNA inside the nucleus. So the DNA and the histone condenses, the chromatin condenses into the chromosomes. It's a whole lot easier to move them together. Now, each chromosome is going to pair up with its counterpart, or its sister, if you will. But we have in the human body, we have 46 chromosomes, 23 pairs. We have 2 chromosome 1s, 2 chromosome 2s, 2 chromosome 3s, and so on up through the 23rd pairing. The chromosome 1s join together and are called sister chromatids. Now we'll have 2 sets of each version, because remember, we've copied everything. So we have 2 chromosome 1s and an additional set of 2 chromosome 1s, because we have to deposit 23 pairs of chromosomes in each daughter cell. So you got to have 2 sets of 1, 2 sets of 2, and 2 sets of 3. When the chromosomes are paired together, they're called sister chromatids, and they are held together at a structure in the middle called the centromere. While all this is going on, we're busy generating spindle fibers from the centrosome. The centrosome, the cell center, these little centrioles are busy generating spindle fibers now, starting that process. As prophase continues, we see the breakdown of the nuclear membrane. We see the spindle fibers now attaching to the sister chromatids at a structure called the kinetochore, and we start dragging the sister chromatids to the middle of the cell. So first they condense, and we start seeing spindle fibers form. Then the nuclear membrane disintegrates, and then we start dragging the paired chromosomes to the middle of the cell. And that leads us into metaphase. In metaphase, we line up on the middle of the cell on the metaphase plate, what we call the cell's equator. So the equatorial plate, the metaphase plate, the middle of the cell. And the reason we do that is very simple. It's to make sure that when we divide, when we physically move the chromosomes to opposite ends of the cell, we're gonna do it evenly. We're gonna make sure that, you know, a complete set of chromosomes goes to the left side and a complete set goes to the right side. And we don't, you know, we're not dragging along a third chromosome 1, you know, on one side and only leave one chromosome 1 on the other side. We don't want to make that mistake. We don't want to have 25 pairs on the right side and only 21 pairs on the left side. I think that's math is right. So we line up on the middle of the cell so that when we start to pull the chromosomes to opposite ends, we can do that. We can do it without making any mistakes. We want to get 23 pairs one side and 23 pairs on the other side. That leads us into the shortest phase of all. It's called anaphase. As soon as those chromosomes start moving, it's called anaphase. As soon as they stop moving, anaphase is over. We drag them to the opposite sides of the cell. Cytokinesis begins in this stage. We actually start squeezing the cytoplasm in this stage. As soon as they stop moving, it's over. Anaphase is over. You have all 23 pairs on either end of the cell. And we're also the spindle fibers that didn't connect to the chromosomes are pushing the cytoplasm apart, because you got to have the cytoplasm in both cells. So we continue to push both sides. We push back and we push the cytoplasm on both sides. So we have all the cytoplasm and all the organelles that each daughter cell will need. Push it apart. Once we stop pushing, we are in telophase. Telophase starts when the chromosomes stop moving. And in telophase, we will see the chromosomes will begin to unwind. The nucleus will reform, the nuclear membrane will reform, the spindle fibers will disappear, and cytokinesis will be finished, and we'll have two distinct daughter cells. So when the movement of the chromosome stops, that is the beginning of telophase. And when telophase is over, we have two new nuclei on either end of the cell. Now, if we didn't have cytokinesis, we would stop there. We would have one big cell with two nuclei in there. Probably be non-functional. But cytokinesis does start in anaphase and continues, and we're squeezing the membrane. We're squeezing the membrane, squeezing the cytoplasm, so we're going to pinch off each daughter cell. And what's causing this is a protein called actin. Actin is a microfilament. It's one of the proteins that makes up our skeletal muscle, and it squeezes like a string wrapped around a tube, like one of those balloon animals. You know how you twist them and they form little tubes and sections. They look like, you know, sausages or something, or sausage links. Well, this is what's happening here. We're squeezing and pulling in here and getting tighter and tighter. We form what's called a cleavage furrow. It looks like this as the cytoplasm is being pinched in half, and we end up with two distinct daughter cells. The ability to do this, the whole process of cell division, mitosis and cytokinesis, is controlled very tightly. There are a variety of go and stop signals that we see taking place here. Why does a cell have to undergo mitosis and cytokinesis? It's because it keeps the cell from getting too big. The bigger the cell is, the harder it is to get nutrients into it, and the more difficult it is for nutrients to get in and waste to get out, and for oxygen to diffuse in and CO2 to diffuse out. Because we have something called surface to volume ratio. If we have a large cell, the surface can't keep up with the contents. The surface area of a large cell is not adequate for enough nutrients to come into the cell. the cell's too big. You can't feed the cell properly because there's just not enough area for nutrients and gases to come in. You can't just, the cell would starve. One great big cell would have a very hard time feeding itself because there just aren't enough spaces in the surface of a large cell to feed the contents inside. So what we do is we keep the cells at a very small size, so the surface area of each membrane is adequate to provide the nutrients needed, the gases needed for the cell to survive. That's surface to volume ratio. So when a cell starts to grow, it will decide at some point, or if it's worn out, it will decide to reproduce. It will undergo mitosis and cytokinesis. Things affect this. Growth hormone drives mitosis and cytokinesis. Bone cells grow during puberty. We start growing. We achieve our adult stature. Our bone cells grow. Our muscle cells will enlarge. They won't grow. We won't make any new ones, but they will enlarge. But our bones will make more and more bone cells to keep up with the enlargement of the muscle cells. Our body gets larger in size. So, growth factors come into play here. We replace worn out cells. We repair or damage to like bone or to burns or to cuts or any kind of physical trauma. Those are the go signals. And they're important. We have to have the go signals telling us when to undergo mitosis and cytokinesis. Stop signals are also important. Stop signals keep us from growing over top of each other. We don't like that. Cells experience something known as contact inhibition. When they come into contact with another cell, they will stop growing and they will stop undergoing mitosis and reproduction because the presence of another cell adjacent to them keeps them in check. All those simple columnar cells that line the small intestine, are adequate are just the right amount for what we need there. We don't overgrow that area because uncontrolled growth is what we define as cancer. Cells experience contact inhibition. They don't like to be crowded, just like humans. We don't like to be crowded either. Even before social distancing was popular, we still had our own personal space. Consider this. You get into an elevator and you're in there by yourself. So what you do, most of us usually stand in the middle of the elevator, the middle of the floor of the elevator. If someone else comes on, typically most of us will end up going, we'll go into one corner and the other person that comes on will go into the other corner and just sort of stand there and not make if you don't know them, not make any kind of eye contact. And if a third person comes in, they're going to stand in the middle near the door so that everybody keeps their personal space. It's only when we start getting a crowd in there that we start getting closer and closer together. We don't like to be crowded. We experience our own form of contact inhibition. Don't touch me, keep my space. Long before social distancing ever was a concept, we still maintained our distance. Nobody likes to be crowded in a line. Whether you're at Food City, whether you're at Dollywood or whatever, nobody likes to have someone jammed up right behind you, practically touching you or even touching you. We don't like that. Cells don't like it. So cells maintain contact inhibition, it keeps them from growing over each other. Now, a couple of proteins come into play here too. We have something known as cyclins. Cyclins are a protein that are manufactured during the interphase part of the cell cycle. Now, they don't become active until they're turned on by an enzyme called cyclin-dependent kinase. Clever name. So they activate the cyclins. And this is not an unusual sequence here. our bodies use many examples of where a protein is formed, but is inactive till another protein turns it on. We use this multi-step process to activate a protein. For example, in our blood, there is a protein called fibrinogen. Fibrinogen is the clotting protein, but it's in liquid form. It doesn't get activated and turned into fibrin, the protein that actually forms a clot. It doesn't get activated and turned into fibrin until another protein called thrombin is formed. Thrombin activates fibrinogen and turns it into fibrin. And just to be extra safe, thrombin is kept inactive as prothrombin. So we have a multi-step process here to turn on fibrin to form a clot. Controlling the cell cycle, we have a two-step process, at least a two-step process here, where we make the cyclins, but we also make the enzymes that turn them on. We can't have mitosis if we don't turn on the enzymes that allow it to happen. And the key for turning on the enzymes is the combination of the cyclin and cyclodependent kinases. So we use these complexes, the cyclins and the cyclin-dependent kinases, to turn on the rest of the enzymes so mitosis can occur. If we don't have cyclin, then nothing happens. So cyclin becomes the key here. So this is how we can keep everything in check. We also have a series of checkpoints. We keep things in control with a series of checkpoints. The most important checkpoint is the G1 checkpoint. And the checkpoint, we have to get through the checkpoint before we can go anywhere else. The cycle can't continue if we don't get past the G1 checkpoint. Remember when we had the orange fencing up last year, and we all had to pass through these checkpoints to be able to go into the buildings because we had to have a mask on, we had to have our temperature checked, and we had to be asked if we had been exposed or were feeling sick or had a sore throat or a fever or whatever. In phase 1 of COVID. That's a checkpoint. Our cells have checkpoints. The G1 checkpoint, we don't get to go into the S stage and copy our DNA if there's something wrong with it. If there is something wrong with our DNA, if the DNA has been damaged, we are very much aware of the status of our DNA. If the DNA is damaged, we stop. The cell stops. Interphase continues, but we don't go past G1. The cell's busy doing cell things, but it's not going to reproduce, at least until the DNA problem gets fixed. The DNA has to be perfect before it can be copied. If there's damage to it, we have to repair that damage, or we have to destroy the cell, one or the other. So the G1 checkpoint is the most important checkpoint. The second checkpoint occurs at G2. Has the damage been fixed? Has the damage been fixed? And we have all the enzymes and proteins and organelles that we need. Well, we shouldn't be getting past G1 if there's damage. We shouldn't get to G2 if the damage wasn't fixed. And the last stage is the M checkpoint, the metaphase checkpoint, essentially. Are we going to line up and send equal number, send the right number of chromosomes to each side? Are we going to have 23 pairs going to each side? If we don't, then we stop there. But the number one checkpoint is the G1, because if we have damage to the DNA for whatever reason, and there are lots of things that can damage the DNA, we stop the cell cycle, we stop the movement from G1 into S until we can fix the DNA. So G1, right over here, number one checkpoint. We continue to process through here. We keep looking for any damage. Did it recover? Did it repair? We check over here at G2, is the damage fixed? Have we replicated the DNA properly? And then do we check here to make sure that the chromosomes are going to get pulled apart equally. What can cause damage? Well, damage to a DNA strand is called a mutation. And usually a mutation involves substituting the wrong base at the wrong location. We substitute the wrong base, or we cut out the correct base and put, what will happen, we can put in the wrong base, that's a substitution, or we can slice out a base and the whole DNA strand slides up one or two. That's really nasty when that happens. So we have a couple of things that can happen. So something can damage the DNA. Radiation is a good example. Ionizing radiation like gamma rays, X-rays, alpha particles, and beta particles exposed to our cells can damage our DNA. Ultraviolet radiation from sunlight can damage our DNA. Sufficient exposure to large amounts of ultraviolet radiation can, over time, damage the DNA in our skin cells and lead to a skin cancer. Smoking can cause DNA mutations. Not the nicotine, but all the other chemicals that are formed in cigarette smoke that are called mutagens. Things like the cyanides and the arsenics and carbon, the benzoapyrenes, all the myriad of chemicals that form when we're burning tobacco and other substances in cigarettes. And those chemicals can cause changes in the DNA, and that leads to lung cancer. Smoking is the #1 cause of lung cancer. And so it's caused by, altering the DNA of the simple squamous cells inside the lungs. Pollutants, air pollutants, water pollutants can cause various types of DNA mutations and lead to cancers. Also, consider the fly ash in the TVA spill 4 or 5 years ago. It's in, was it Blount County, I think, or Anderson County. It's over somewhere in the Oak Ridge area. You're all familiar with it, I'm sure, about the big spill that happened the day after Christmas 5 or 6 years ago. Fly ash is what's produced when you burn coal. It is the residue left behind from burning coal. And if it's not stored properly, it can pose a risk to human health because it contains metals, it contains chemicals from the burning of the coal, that are dangerous if exposed in high quantities. Well, a lot of people in the cleanup crew that worked on that event after that big massive spill and have ended up developing various types of cancers, from different chemicals, in the air or stuff they've ingested. Chemicals themselves, if you're exposed to chemicals, if your patients are exposed to chemicals, if they're a farmer, and farmers use fertilizers, farmers use pesticides all the time. Some of the components of those chemicals can cause damage to the DNA. If a person works in a factory and they're exposed to certain industrial chemicals, it can also cause mutations to their DNA. And then, of course, we have viruses. Human papilloma virus is the virus that causes cervical cancer. And cervical cancer is, I think, the #3 killer around the world in women. And it's caused, usually caused, let me rephrase this. one of the primary causes of cervical cancer is this human papillomavirus, and the virus damages the DNA. Viruses, not every virus causes cancer, but a lot of viruses work by damaging the DNA of the host cell or taking it over. Because remember, all a virus wants to do, is, if it were alive, because viruses are not alive. A virus is nothing but a strand of DNA or a strand of RNA wrapped in a protein coat. It binds to the surface of a living cell. It injects its DNA into the cell, and the fragment of DNA from the virus incorporates itself into the healthy cell's DNA, replaces sections of the healthy cell's DNA, takes the cell over, and all it does is make copies of itself. That's all a virus does. It makes copies of itself until the cell explodes. It fills up the cell with viral particles. The cell explodes, and then those viral particles are strewn out throughout the body or breathed out, with air, or come out with saliva and infect other cells. And eventually, you damage enough cells and you become symptomatic, and if you kill enough cells fast enough, you die. So those are things that can affect the DNA. If we can't stop that problem, then if a cell is exposed to any of these conditions, there are only a few options. The first is to for the cell to repair itself. That's its best option, is to repair itself, if it can. But if it can't repair, we do have a mechanism to try and repair any kind of damage. If we can't repair it, we have three things can happen, none of them good. If the cell can't repair the damage, it goes dormant. We call it senescence. It's irreversible. The cell doesn't disintegrate, it just stops right there. The second thing a cell can do is undergo self-destruction. Apoptosis, programmed cell death, self-destruction, cell suicide. I prefer self-destruction over that. But apoptosis, lots of cells will undergo apoptosis if they are damaged beyond the point of repair. The third condition, which is probably the worst, is the cell can start experiencing uncontrolled growth, leading to the formation of a tumor, which may or may not be malignant. It may be a cancerous growth, a cancerous tumor. Some tumors are non-malignant. They're a benign tumor. They're a growth that doesn't harm us. Some tumors are malignant, and they're a cancerous cell, and they're going to spread their cancer cells out to other parts of the body. And if not caught in time, it can kill the patient. Apoptosis, programmed cell death, self-destruction, is caused by results from the lysosomes within the cell releasing their digestive enzymes to break down the cell membrane and break apart the cell. And at that point, after the cell is broken apart, then all the neighboring cells and white cells will come in, eat the fragments, and eliminate the dead cell. Here we see something known as this bulging out here. This is a cell magnified 8,000 times, and we see these bulges. That's called blebbing. When the cell membrane blebs like that, it occurs because the cytoskeleton has been dissolved. And this indicates to the cells around it that that particular cell is damaged and is destroying itself, and it's going to break apart into small fragments, and let's go eat it. So, which is what's happening over here. Here is a white blood cell, and it finds a cell fragment from an infected cell, and it destroys it. It takes it in, engulfs it, and destroys it. So this is what's known as a macrophage, a large cell that eats solid particles. It all started because we had a healthy cell. There we go. There's an in the animation. The healthy cell was infected by a virus, and the cell can't repair the damage caused by the virus and will start breaking itself apart into fragments, which are then eaten by the macrophages or the neighboring cells too. So this is how the cell can ultimately solve its problem through apoptosis. Okay, I'm going to stop here at tumor suppressor genes because I don't have enough time to really go into this like I want to. And we will pick up with this on Monday. So, okay, let me get out of here. And I will see you all later. So, okay, thanks a lot and have a good day.